Assalamualaikum and greeting everyone. Welcome to my proposal presentation. I'm Nurdini Afiqah binti Suhaimi. I will presenting my final year project entitled Investigation of the Antibiofilm Potential of Ascorbic Acid against Staphylococcus aureus under supervision Dr. Fakharul Zaman, Raja Yahya. This is my presentation outline. What is the biofilm? According to the Wikipedia et al. 2021, biofilm is a population of bacteria attached to biotic or abiotic surface that are encased in extracellular matrix and play a crucial role in pathogenesis. Mermier and Berry state that extracellular matrix contain protein, nucleic acid, polysaccharide, and lipid that wrap cells in a sticky matrix, increasing their chances for survival in adverse conditions. They also mention S. aureus is one of the bacteria that form biofilm and cause persistent infection. Next is problem statement. S. aureus is responsible for a wide variety of infections due to its ability to coordinate the production of many relevant factors. A Fakharul attribute of S. aureus is its ability to form biofilm, which are crucial in the etiology of life-threatening infections such as endocarditis, bloodstream infections, pneumonia, and infections connected to the implant. For your information, antibiofilm activity of ascorbic acid against S. aureus has previously been investigated. However, its efficacy at different time intervals remain unknown. The objective, first, to determine the effect of ascorbic acid on the biomass of S. aureus biofilm at 24 hours, 48 hours, and 72 hours. Second, to determine the effect of ascorbic acid on the surface coverage by S. aureus biofilm at 24 hours, 48 hours, and 72 hours. Significance of study. This study is very important to know the effectiveness of vitamin C inhibiting Staphylococcus aureus biofilm. This study also helped to treat the disease caused by the biofilm. For research question, is ascorbic acid effective against Staphylococcus aureus biofilm? Now move to the literature review. Firstly, about Staphylococcus aureus. Grace and Fetch mentioned that S. aureus was first identified by Scottish surgeon named Alexander Oxford in 1818. Exley et al. State that S. aureus is a gram-positive bacteria and has cocci shape. S. aureus' ability to form biofilm and it causes infections like endocarditis, bacteremia, sepsis reported by Silva Santana et al. Secondly, the stage of biofilm development. 2022 study it have 5 stages of biofilm development. Firstly, initial attachment when single free-floating microbe land on biotic or abiotic surface. The next type is irreversible attachment, where microbial cells gather and attach. Then, maturation 1 occur when the microbial colony grow and divide for biofilm formation. Next, maturation 2. Biofilm form and may develop spatially well arranged. Lastly, the dispersion stage. Microbe from section of biofilm disperse and release free-floating microbe for further colonization. Next, ascorbic acid. Ascorbic acid, also known as vitamin C, was first identified by Albert Stein-Gorey in the 1920s. It cannot be synthesized by human but can obtain from dietary supplement. Price et al. Show ascorbic acid has antimicrobial activity against S. aureus. Vitamin C also suppresses the synthesis of extracellular polymetric compound, a central component of biofilm, hence disrupting the biofilm structure. Vitamin C can alter and improve both innate and adaptive immune response. So, now for the methodology, regarding the flowchart, firstly, preparation of Staphylococcus aureus. After that, verification of S. aureus by gram staining and biochemical test. Then, antibiofilm test will be split into 2 parts, which is biofilm biomass assay and biofilm surface coverage assay. First method is the preparation of S. aureus by conducting a streaking plate method on nutrient agar. Then, transfer the colony into the colonical flask to grow the bacteria. The second method is verification of S. aureus by gram staining and biochemical test. The function of the biochemical test is to identify bacteria species by differentiating them based on biochemical activities. This experiment will be conducted 3 types of biochemical tests, which is coagulase test, catalase test, and novobiasin susceptibility test. For gram staining, it is to distinguish between gram-positive and gram-negative bacteria by coloring the cell red or violet. The third method is antibiofilm test. It will be split into 2 parts, which is biofilm biomass assay and biofilm surface coverage assay. For biofilm biomass assay, use 96 well microplate and 6 different concentration of ascorbic acid. Then grow S. aureus overnight in tryptic soil broth, which is TSB culture media at 37 degrees Celsius, and then add 100 microliter of bacterial suspensions to 100 microliter of 100 microgram per milligram ascorbic acid in 7 wells. Then repeat the step with the different concentration, incubate for 24 hours, rinse 96 well microplate twice with the phosphate buffered saline, then stain with the crystal violet for 5 minutes, wash the plate and allow to dry. After that, solubilize in 96% of ethanol and read the optical density at 600 nanometer. Repeat for the different incubation time for 48 hours and 72 hours. After obtaining the result, do the statistical analysis. T-test will be performed to determine significant difference of a p-value smaller than 0.05 between control and test group of biofilm biomass assay. For biofilm surface coverage assay, firstly, grow as aureus overnight in tryptic soil broth culture media at 37 degrees C, and then glass cover slip will be added to each of a 6-well polystyrene microtitle plate. Then 2 ml of bacteria suspension mixed with the 2 ml of ascorbic acid in 6 different concentrations. After that, discard and remove the cover slip carefully. Last but not least, the result will be analysed using ImageJ program to see the surface coverage after inhibition by ascorbic acid. This is my Gantt chart. This is my references. That's all from me. Thank you.